Next topic, which is cardiac muscle and how its action potential takes place. So here we're focusing on the action potential of cardiac muscle more than the other properties. Regarding the other properties of cardiac muscle, we'll have a special session on that when we do cardiovascular system in more detail. That time we will come back to this, and there will be lots of references we'll make over here, but for now we'll just focus on the electrical activity of the heart and how it works, okay, and a little bit about the cells. So if we look at cardiac muscle, they are striated just like your skeletal muscle. However, they are not voluntary muscles. Remember, only your only voluntary muscles are the skeletal muscles. Then you have the fact that they are mononucleated, so they are single nuclei. And so if we look at this, you have the Z lines, and between the Z lines, you have the sarcomere. Each sarcomere has a little nucleus, and this is your cardiac cell over here. So they have a single nucleus. If you look at a skeletal muscle, however, you had this big muscle over here, and you had a sarcomere extending from one end to the other end. The Z lines, well, these are like individual cells that fuse together, so you get this multinucleated cell in the skeletal muscle. Right, so here we have the mononucleated cells. They're quite long. If you look at the fibers over here, branching over here, branching. Okay, they branch a lot. This is important. And like we said, the nucleus is in the center of the cell, and they have a lot of myoglobin because these cells, the heart cells, are extremely sensitive to hypoxia, so the myoglobin holds some oxygen in reserve if required under times of stress. So it's very important to know that this is really rich in myoglobin. And the other thing is, the cardiac muscle has something called intercalated discs. So what are these intercalated discs? They connect one cardiac muscle to the other cardiac muscle end to end. So if we look at this picture over here, it takes an irregular step-like path. Can you see over here where the arrow is pointing? It's like steps, but they are quite irregular, like a jagged face of a mountain in a way. And here you have three components in it. Number one is fasciae adherens. Okay, fasciae adherens. So this is in the transverse portion of the intercalated disc, and it binds the muscle from end to end, like your adherens junctions, which we will talk about in the epithelium chapter in general physiology. So this adherens junction, it helps in attaching cells and one cell to the next cell, like in epithelial cells. So you have these epithelial cells next to each other. You have something called tight junctions, adherens junctions, desmosomes, and hemidesmosomes that link the epithelium to the basement membrane. But anyway, we'll talk about that later. The point you should know about is that you have these adherens junctions in the epithelial cells. Then we have gap junctions. Now, what are the gap junctions? They're in the lateral portions of your intercalated discs, and here you have these gap junctions that again help in connecting one cell to another. So you have these intercalated discs that connect one sarcomere to the other, and what happens is you have these channels called gap junctions. They're made of connexons. So connexons, 6 connexons from one cell that make a gap junction on one half will combine with 6 connexons on the other cell. So 6 on one side and 6 on the other side for these connexons will make up a gap junction. Now what this does is this connects the cells, and at the same time you have this sort of an ion channel, meaning a doorway between two different adjacent cells. And since the cytoplasm of the cells are now connected, think of a window connecting two adjacent rooms together next to each other. So what happens is you can easily hear what happens in the next room, or if somebody sprays a nice room freshener in one room, the smell will eventually make it to the other room. So that means to say gap junctions allow for communication between the cells, allowing for something called functional syncytium. That means if one cell, for example, depolarizes or gets an action potential because some cations are coming inside the cell, then what happens is these cations can diffuse to the other cell via the gap junctions. and this allows for the action potential to continue in a wave-like fashion and spread throughout the entire surface, in throughout the entire heart muscle, the myocardium. So the gap junctions are very important over there. Then you have the desmosomes. This is again a part of the intercalated discs, and they play a role in, they are present in both transverse and lateral portions, and they play a role in connecting the cells over here. Okay, so desmosomes, they're again, you can refer to the epithelial chapter and the connections in cellular biology section, and we'll be doing that very soon. So now we have the other thing over here, which is if you look at the the cardiac muscle, look at how the mitochondria are arranged around the nucleus over there. They make a nice ring over there. Just be aware of that. And these are your intercalated discs, and see the branching pattern over here. Some other different unique features of cardiac muscle are they do have T tubules and L tubules just like your skeletal muscle, but you should be aware that they're not organized the same way as in the skeletal muscle. So in the skeletal muscle you had something called triads, if you remember, where you remember the AI junction, there was a T tubule over here and a T tubule over here. So at the AI junction, then there were L tubules on both sides, L tubule, L tubule, L tubule. So you had the triad over here and another triad over here, one T tubule and two L tubules in each of these sarcomeres. Well, in your cardiac muscle, you don't have two of these triads in each sarcomere. You have one single dyad, D-I-A-D, dyad, dyad. See, dyad. So what is a dyad? It's one T tubule which is located at the Z line of one sarcomere, and the L tubule that extends throughout the entire length, which releases the calcium. So one T tubule and one L tubule will make up a dyad. There's no foot process over here like in the skeletal muscle. Remember, there was a connection between your dihydropyridine receptor and ryanodine receptor. So what is this dihydropyridine? I recommend you watch the excitation contraction coupling chapter, but I'll just mention it. So remember how an action potential forms in the cell membrane, and then to bring that action potential deep into the cell membrane, we have the T tubule. So if an action potential forms over here, it goes down the T tubule, and then it causes a change in the T tubule. There's a special sort of an ion channel called dihydropyridine receptor. Dihydropyridine, DHP, which is written over here, dihydropyridine receptor. You can see in this red box over here. So this dihydropyridine receptor is sensitive to the positivity that's coming in the action potential that has come down over here. So there's an action potential that's coming down. Cations are coming inside over here, so most likely sodium during phase 0 of the cardiac action potential. We'll talk about that. This makes the cell positive on the inside, and the dihydropyridine picks up on this signal, and then it is going to, in the case of cardiac muscle, cause a change in ryanodine receptor by spraying calcium. It sprays calcium, so calcium is sprayed by the dihydropyridine. You see there's this ion channel over here, can you see, which is linked to the dihydropyridine. Well, so the dihydropyridine is going to open the channel, calcium is going to get sprayed on the ryanodine receptor, RYR, ryanodine receptor. And this ryanodine receptor, by the way, we call this dihydropyridine and ryanodine receptor because they are blocked by those chemicals called dihydropyridine and ryanodine. Alright, so anyway, so dihydropyridine releases the calcium, and it will now bind to ryanodine receptor. This ryanodine receptor is going to open its channel because calcium was sprayed on it, and it's going to release even more calcium, which is stored in these L tubules thanks to the Cerca ATPase pump. Alright, so Cerca ATPase pump is located along the length of the L tubule, and it really concentrates and takes the calcium actively. Takes a lot of uses a fair amount of energy in the form of ATP to get this calcium concentrated over here. So this calcium gets released, then causing the excitation contraction coupling to take place in the muscle. And so remember, here it is calcium induced calcium release that is taking place for releasing. In the skeletal muscle, what happened was dihydropyridine had a physical connection with ryanodine with something called the foot process. Again, watch the video. Right? So I want to talk about one more important thing, which is this chemical called phospholamban. So, how does this work? Well, look, circa ATPase causes relaxation of the muscle, of the cardiac muscle, by pumping the calcium back into the L tubules. No calcium in the cytoplasm means no excitation, contraction, coupling. So, what happens over here is the if we can make circa ATPase work faster, you can relax the muscle more efficiently and much faster, causing a stronger, better, a faster contraction of the heart in the next beat. Okay, so listen to this. It sounds counterintuitive, but if we get rid of calcium from the cytoplasm quickly in the cardiac muscle, we should be able to actually make a more efficient contraction to take place, more efficient contractions of the heart. So what happens is there's this chemical called phospholamban over here. Phospholamban inhibits circa ATPase, doesn't let it work too much. And if we look at the heart, it's innervated by your autonomic nervous system, and you have sympathetic nerves and parasympathetic nerves over here. Parasympathetic via the vagus nerve, and sympathetic is through these intermediolateral gray horn cells, meaning the cells that come from the spinal cord. They're sympathetic nerves. We'll talk about that in its respective class on autonomic physiology. The point is, norepinephrine gets released from these sympathetic nerves, and it binds to G protein coupled receptors, GPCRs, on the heart, like your beta 1 receptors. Those are the ones which are present on your heart, and that causes the increase in CAMP levels, cyclic adenosine monophosphate levels. This is a second messenger. Again, we're going to have a class on all of this intercellular communication soon. Now, the CAMP activates protein kinase A, and this is an enzyme that is going to put a phosphate group on something else. That's why it's called a kinase. A kinase is an enzyme that phosphorylates. It puts a high energy phosphate bond onto some molecule. So we have this phosphate group, it sticks it to phospholamban, and this is going to inhibit phospholamban. So no more phospholamban working means no more inhibition of circa ATPase. So circa ATPase is got a boost in its effectiveness, in its ability to take calcium back into the cell. So this is what happens over here. You get this phospholamban, which is inhibited because of the norepinephrine, because of sympathetic activity. Right? And the other things you should know over here about cardiac muscle is myosin heavy chains have got alpha and beta, are of the alpha and beta variety. So there are different types of myosin heavy chains. One is alpha, one is beta, and the alpha chains are more present in the atrial muscle. There are beta also in the atria, but alpha predominates. Alpha myosin heavy chains predominate in the atria, but the beta myosin heavy chains predominate in the ventricles. Though you do have alpha myosin heavy chains in the ventricles also, but predominantly alpha in the atria, beta in the ventricles. See, A alpha. So that's how you can remember it. Now, in basal conditions, you should know that your heart, it takes its energy a little different from other tissue. Most of the energy, 60%, comes from fats, 5% from ketone bodies, and 35% from carbohydrates. So fats and ketone bodies and amino acids, they constitute a fair amount of the energy. 35% is carbohydrates. So 60% fat, that's the most. 35% is carbs, and the last one is ketone bodies or amino acids that use up the energy, that give the energy for the cardiac muscle. Now we're going to talk about cardiac action potential. And if you look at it, if I recommend you look at the video on the action potential in neurons action potential. Remember, there was an RMP, there was a stimulus artifact, local response, spike potential, repolarization, hyperpolarization, RMP. Watch that video. It's very important to understand stuff. So assuming that you've watched that video and you understand the concept of depolarization as positive cations coming in the cytoplasm, and the cytoplasm getting more positive. Repolarization as the cytoplasm becoming more negative, meaning the inner part of the membrane becoming more negative. We'll now go into what is cardiac action potential. So When we speak about cardiac muscle, they have something called fast response action potential, and there's another type of tissue in the heart which has action potential, and that is your pacemaker tissue. So the pacemaker tissue has its own action potential, and its shape is a little different from cardiac. You can see over here. Those are called. They have something called slow response action potential, and there's something called pacemaker potential. We'll be stressing on later. First, I want to do cardiac action potential. This is an important concept, and so let's get into it. If we look at your cardiac action potential, you can number 5 phases, but they are numbered 0, 1, 2, 3, and 4. Okay, 0, 1, 2, 3, and 4. This is 0, the spike. Then you have 1 over here. Then you have 2 over here. And you have 3 over here. And 4 over here. So what is happening over here? Okay, before we go into this, just remember that your heart, it has got pacemaker tissue, SA node.